Dr. Jaffe, what testing do you recommend for learning whether or not someone is truly gluten intolerant?
There's probably no more confused and confusing subject in discussion commonly today than gluten sensitivity. Now, depending on how you look at the question, you have very different understandings of what intolerance means. True gluten intolerance is best measured immunologically with a lymphocyte response assay, such as the LRA by ELISA Act. And since LRA by ELISA Act has less than 3% variance on blind split samples, it's the most sensitive and repeatable, reproducible assay. We recommend LRA testing if your question is gluten sensitivity, or what immunologically is called intolerance. But now let's talk about people, because grains like wheat, oats, and other gluten-containing grains, and by the way, anything you can make a bread out of has something sticky in it called gluten or gliadin, and wheat gluten is a little different than oat. And then another aside, and I think this is an important point for consumers, I recently read a very careful study that found that 50% of the products that are labeled gluten-free contain gluten. And the importance of that is that gluten is hard to digest. Grains are hard to digest. So what if you just have maldigestion and dysbiosis and maybe some atrophy of digestion called enteropathy? If you have weak digestion, if you want to rebuild your digestive competence, our recommendation is avoid all grains, also avoid cow dairy and meat, because they're hard to digest, and very often they're contaminated with whatever the animal was fed or however the grain was grown. Most of the wheat today contains glyphosate. That's an issue. I would say plant-based is a good idea, but with lots of seasonings, spices, and herbs, as much as possible organic or biodynamic or locally grown, so you know the farm to fork or farm to table connections. And so I often suggest to people who are negative immunologically, but who are strengthening their digestion or restoring the competence to their digestion, that they should have grasses, fruits and vegetables, herbs and spices, seeds and nuts, and reduce as much as possible. And in my kitchen, essentially, we've reduced 100% the processed foods. We don't even use edible oils because we get plenty of oil from seeds and nuts and whole foods like avocados. And we want to know where the food comes from. And then we find that we can prepare it easily, whole, and tune the flavorings to our preference using herbs and seasonings and spices. And I often use mustard seeds or sesame seeds in whatever we're putting in a tagine, which is a way of simmering and preparing foods easily. And I recommend high fiber foods, lentils and beans, garbanzos, chickpeas, grasses, wild rice, such a delight, and other of the easy to digest grasses. Now back to the gluten question. The reason that people have tried to do genetic risk testing is because the gluten issue is complicated and commonly misunderstood. And if you want to get more confused, do the genetic testing. And here I'm going to quote Eric Lander. This is the man who sequenced the mouse genome while NIH was sequencing the human genome. He ran the Office of Science and Technology Policy for President Obama. He's back at MIT now. And what he says is, unless you measure the entire genome and the RNAs, because RNA is as or more important than DNA, and transgenerational influences on RNA, and epigenetic environmental influences on the DNA, causing changes in the DNA for good or for ill, based on your habits of daily living, based on epigenetics, which is where we place emphasis. Eric says, if you do a SNP, if you just look for a specific gene, and you think that it's a blueprint, you think it's fixed, You don't understand genetics. Genetics is really complicated. I'm old enough to remember when reverse transcriptase, for which they gave David Baltimore the Nobel Prize, was so controversial that for years he had to keep at the research until it was obvious to everyone that he was right, that reverse transcriptase repairs the DNA, and there are other molecules that repair the DNA because the DNA has been subject to damage as long as there's been DNA, and the body is smart enough to have repair molecules which turn out to be dependent upon antioxidants, buffering minerals for their manufacture and function. And then we have Jerry Trier. Now, this is the man who really put celiac on the map. And he came to the conclusion that if you really want to diagnose celiac or gluten sensitivity, which is often confused, they're not the same. But if you really want to diagnose someone as celiac or not, you have to do a small bowel biopsy and an electron microscopy to see at the ultrastructural, tiny, tiny, tiny level whether there has been certain kinds of damage to the upper small intestine. And then he pointed out and published repeatedly that gluten is not the only cause of celiac disease. Yes, 2/3 or more of people who are biopsied are gluten sensitive, but then there's, and that would used to be called tropical sprue for whatever reason, and then there's what he coined the term, or Sheila Sherlock coined the term, non-tropical sprue, which means you're sensitive to something other than gluten, but it's giving the same damage because of immune intolerance, immune hypersensitivity. And part of the reason that I developed the LRA by ELISA-ACT test in the early 1980s was to make clear the difference between immunologic intolerance, hypersensitivity, which means sustained avoidance for at least 6 months if it's a strong reaction, while you're restoring and strengthening digestion, in contrast to simple maldigestion or poor digestion, meaning you should digest or consume and digest the foods that you can easily assimilate and eliminate without immune burden based on the LRA testing. So if the LRA test is negative towards gluten or gliadin or wheat, that's not saying that it will help you to eat that food, and it's not saying that you're really functionally okay with the food. It means you're not immunologically hypersensitive, which is a more advanced form than simple maldigestion. Although weak digestion goes along with partially digested food remnants that act as foreign invaders coming into the body, flooding into the body through the impaired intestinal wall, and usually the same person has a lack of protective mucosal barrier. There's secretory IgA that is plentiful in healthy people, but the most common acquired immune deficiency in America is now lack of IgA or secretory IgA, sometimes called SIGA, for the acronym secretory IgA or SIGA. Mucins that are designed to trap partially digested remnants and then let the healthy bugs, notice you have to have lots of healthy bugs, these are the probiotic organisms, break down the food to building blocks, which the body can take in and recycle. Now, one additional point, the body has a capacity to take in and recycle a small amount of foreign invader because in the body, we have cells that have what's called a lysosome. Think of that as the recycling center of the cell. If anything comes in that's foreign, immune cells are there to gobble it up, put it in that lysosome recycling center, break it down to the elemental building blocks, amino acids and sugars and maybe some fats, and then the body can be benefited. So complicated question, not a simple answer. However, do not, in my clinical experience, do genetic testing for gluten intolerance because the results will vary over time. They're not fixed. The notion of there's a widespread misunderstanding that if you come from the parts of the world that didn't have wheat, you won't have the genetic capacity to digest wheat. Actually, what happens is the DNA is constantly rearranging Sheila Sherlock got the Nobel Prize for showing that genes jump around, not just rearrange, they jump around. And when we get deeply into the question of genomics and genetics, my conclusion, based on listening to people who know lots more than I, is that if you want to do the entire genome at any given moment in time, it's like a snapshot, but be sure to include the RNAs, and there's different families of RNA. However, if you follow the epigenetic health promoting approach that Perk Integrative Health has integrated, you should improve and change for the better your DNA. But if you don't take in enough protective antioxidants and buffering minerals and essential cofactors, now you're going to be vulnerable because the damage that happens to everyone every day in their genetic code will not be repaired efficiently. And over time, you will have chronic, progressive illness. So the genetic testing is very elegant, it's new, it's in conversation. But when I look at the evidence, when I look at the science, and it's something that I believe I'm equipped to do, but it takes a challenge, even for me, to get behind the headline conclusion to the evidence. And the evidence is follow epigenetics, follow LRA by ELISA Act testing, use something like our health appraisal questionnaire to determine what lifestyle components will help restore and rebuild yourself. And when people talk about genetic testing for gluten, you can be polite and say as little as possible and avoid that genetic testing, but do the functional epigenetic tests, including the predictive biomarkers. That includes the LRA by ELISA Act immune tolerance test and the other essential metabolic fundamentals. And that will give you much more useful information, actionable information, information that will keep you functioning at a young age, whatever your chronology is. You know, my birth certificate tells me how many years I've been on the planet. But I'm glad to say that by most measures, I'm functioning between the ages of 35 and 40. And if I can keep that up for maybe another half century, and I would like to be dancing at 120 and have you with me, I'll consider that a self-experiment of some success. And I'd like other people to put to the test what we have pioneered and advocated for decades, which is epigenetics, not genetics, epigenetics, not genomics, lifestyle, which means epigenetics, rather than snips and snaps and snups.